Hello friends, today I am going to explain you chapter 9 strategies for enhancement in food production part 4 based on NCERT. But before explanation, I remind you please like, share and subscribe our channel and press the bell icon to get the notification of new videos. In this topic, first I have to discuss about the single cell protein. Single cell protein is one of the alternate source of protein for animals and human nutrition. The idea of obtaining vitamins from the microorganism like yeast and mushroom was developed to solve the problem of hunger and malnutrition. Microorganisms are very small. They cannot be seen through your naked eyes. They are capable to produce high rate of biomass production. This biomass is used as food by humans, and these microorganisms are grown in a particular substrate. What is the substrate? Where the microorganisms are grown. The substrate used for the production of biomass, that is like industrial effluents, whey, water of curd, low cost organic material, material and commercial production of SCP is mostly based on yeast and some other fungi. SCP is very rich in high quality protein and is poor in fat. Now one of the best example I have discussed, that is, it has been estimated that 250 kg cow produces 200 gram of protein only per day. In the same period, the 250 gram of microorganism in a petri dish like Methylophilus, Methylotrophus, because of its high content of biomass production and growth, can produce 25 tons of protein. So this is the quality of the microbes, and we have to harvest it from the microorganism, the single cell protein, which is very useful for the human beings. Some of the common microbes, those are SCP producers like cyanobacteria. One of the example I will give you, cyanobacteria, that is Spirulina. And bacteria, that is Methylophilus Methylotrophus, is a bacteria which is responsible for the production of SCP. And yeast is also used in this process, that is Candida utilis, which is used in the production of SCP. Some fungi, that is filamentous fungi like Fusarium graminarium, is used in the production of SCP. What are the most advantage of the SCP? Because one of the important thing, microorganism has a high rate of growth in a very small period of time. They are growing very fast, and we can grow in a wide range of raw material, can be easily harvested. That can be grown in a various raw material and easily harvested. We have relatively high protein content as compared to other healthy food items. It is easily produced in the laboratories. That is one of the more useful advantage of the SCP. Now we have to discuss about the plant tissue culture. What is plant tissue culture? It is a method of growing cell tissue or organ in a nutrient medium under controlled conditions. Like part of the plant, some part of the plant is taken out from the plant and is cultured. So that part is known as EXPLANT. What we call them? EXPLANT. And this is cultured in the liquid, solid or semi-solid medium. The nutrient medium includes inorganic nutrients, organic nutrients and growth hormones. So these are the nutrient which is responsible for the growth of the EXPLANT. And the cells which are taken that are totipotent in nature. Totipotent means that having the capacity to produce an entire plant body. So this is the cellular totipotency, is the ability of plant cell to give rise an entirely new plant. This is called cellular totipotency. This method of producing thousands of plants through tissue culture is called micro-propagation. So in this method, we have to develop thousands of plants in a lab. So this method of producing plant is known as micro-propagation. One of the important characteristics of these plants, they are genetically identical from where the explant is taken. So this is one of the important characteristics, because the same parent, all the explant or the part of the plant is taken from the same parent. We have to develop sometimes a disease-free plant through this technique. Disease-free plant develop, but we have to take in the explant from the meristem. Meristematic cells are always dividing in nature, so that this explant is taken from where they are only. So the disease-free plant we have to develop. And genetically identical plants are also called somaclones. What we call them? Somaclones. Clone means the same genetic constituent from where the explant taken, from where the part of the plant is taken, and we have to develop a different plant bodies that is known as somaclones. Many important food plants like tomato, banana, apple have been produced by using this method. Another one very important thing, that is somatic hybridization. When hybrid is produced by fusion of somatic cells of two species. Somatic cells means the body cells known as somatic cells of the two species is known as somatic hybrids. And the process of somatic hybrid is called somatic hybridization. What we have done in this, we have to take in the two species cells, and cell membrane and cell wall is dissolved, and the protoplasts are fused together to form the somatic hybrid. The protoplasts of two plants are brought in contact and fused by means of chemical. They are not fused alone. They are fused in presence of chemicals like polyethylene glycol and sodium nitrates are used for the fusion of these protoplasts. And fused protoplasts allowed to grow in a culture medium. Soon they develop their own walls called somatic hybrid cells. Like one of the example I will give you, that is tomato is a somatic hybrid. How many plants cells are fused in this tomato? That is, one is potato and the tomato that belong to different genera. The cells of the potato, the protoplast of the potato and the protoplast of the tomato are fused together and form tomato. This process is known as somatic hybridization. Now we have to discuss about some questions related to NCERT, that is, what is the major advantage of producing plants by micro-propagation, and another one question is find out what are the various components of the medium used for propagation of an explant in vitro, what is SCP and its advantage. So these type of questions are coming from this topic, and you have to practice or you have to done at your home these questions. And this is all about the topic single cell protein and plant tissue culture. Thank you, students.